Lots of games we play have some element of randomness, like a roll of a die or a random hand of cards. One game that is often touted as being based purely on the skill of the players is chess. Each move in a chess game is deterministic. The player makes a conscious decision about where to place their piece. This doesn't mean that each decision is good or well thought out, but it is free of the randomness of a dice roll. If you pit two players against each other of vastly different skills, say a grandmaster and someone who just barely knows the rules, the grandmaster is virtually guaranteed to win every time. They can't be foiled by being dealt a bad hand. But imagine two friends, call them Sarah and Tom, who are equally skilled at chess. They meet every Sunday at the park to play chess and have in total played 100 games of chess. Since they are of equal skill, sometimes Sarah wins and sometimes Tom wins. Sarah has a slight edge right now, having won 52 games to Tom's 48. That is, within a small margin of error, very close to just 50-50. In fact, you could accurately predict this distribution with a simple coin toss. Thus, despite the fact that each and every game and all the moves within the game are strictly deterministic, the emergent pattern is no different from random chance. In evolutionary biology, we often like to divide up the mechanisms of evolution into two bins: the deterministic bin and the random bin. The random bin includes things like mutation and genetic drift, while the deterministic bin is for natural selection. Most people think of these bins as finely demarcated, in no small part because creationists like to say things like, "How could humans have evolved randomly?" To which most people respond something akin to, "Mutation is random, but natural selection is not." This implies that selection applies a kind of directionality to the random variation generated by mutation. When it comes to correcting misunderstandings of evolution to creationists, or just generally explaining the basics of evolution to the general public, this is perfectly fine. But if this channel has any concrete goal, it's to challenge my viewers to think a little deeper about these topics, to push a little beyond the basics. After all, if you just wanted to learn the basics of evolution, there's a plethora of other higher profile YouTube channels you could be watching. I like to humbly think you are here for the challenge. In this video, we're going to chat through two ways that natural selection, a decidedly deterministic mechanism, can be thought of as an agent of randomness. We're going to divide these two ways into top-down randomness and bottom-up randomness. And thinking about selection as a kind of random variable in nature, it will help us explain many features of the biological world, such as how genetic diversity is maintained in populations, and even how speciation can occur in the complete absence of morphological differences between populations. Let's get started. The first scientific research project I was ever involved in was during my undergrad. We were doing ecological surveys of ponds on a large swath of land that the university had just acquired that was being converted from cattle grazing to a natural grassland. Our goal was to document the aquatic biodiversity, and we were looking mostly for these teeny little crustaceans called water fleas. Water fleas are super fascinating little critters. As adults, they have a single fused eye in the center of their head, and their bodies are completely translucent, so you can see all their organs, including the eggs that a pregnant female might be carrying in their brood pouch. They get their name from the way they move around in the water, using their large antennae to flick about. They breathe through their legs, which are actually modified to act like gills. Most water fleas are parthenogenetic, which means that males typically very rarely appear in the population, and females mostly reproduce by the production of unfertilized eggs. During periods of environmental stress, females will produce males for sexual reproduction, but it's relatively rare. Some of the biggest and most well-studied water fleas belong to the genus Daphnia. Many species of Daphnia have been studied due to their remarkable ability for what's called adaptive plasticity. This is when, during development, if the embryo detects certain chemicals in the water that are produced by their main predator, midge larvae, they will develop these large spines on their head, so when the midge tries to eat them, it stabs their mouth and makes them spit them out. In Portland Ark, Indiana, there is an isolated experimental pond of Daphnia pulex that has been studied now for over 10 years. Each year, researchers went out and collected around 100 water fleas and sequenced their DNA. This allowed them to estimate how much genetic diversity the little pond had and how their allele frequencies were changing over the years. Natural selection is expected to push alleles in one direction or another depending on the effects of that allele in the given environment. If there are some adaptive alleles floating around, they should increase in frequency over the period of time studied. If all of the alleles are strictly neutral, however, then they should just fluctuate randomly due to genetic drift. Since there were several thousand individual water fleas in this pond, the year-to-year fluctuation of allele frequencies due to genetic drift should be pretty small. Let's take a moment to chat through why. Let's say there are two alleles in a population. We'll call them A and B, and these two alleles are at frequencies P and Q, where a frequency is simply the count of individuals with the allele divided by the total number of individuals in the population. In an infinite population with no selective advantage, the frequencies of P and Q will never change. This is known as the Hardy-Weinberg law. But as soon as there is a finite number of individuals, the allele frequency will change just by chance alone. How much it changes is a function of how big the population is. You can think about it like this. Since an infinite population has no change, populations that are extremely large in size, say approaching infinity, must have very small change on a generation by generation basis. The farther you get from infinity, the greater role chance can play. Mathematically, we can express this variance as the variance in P is equal to PQ divided by 2n, where n is the size of the population. If P is at 80% and Q at 20%, and the population has 5,000 individuals, then the generational variance is expected to be about 0.000016. If instead the population only has 50 individuals, that variance increases to 0.0016. The key takeaway here is that genetic drift on a generational basis is pretty slight. You don't expect to see big shifts in frequencies every generation unless the population is very, very small. Furthermore, drift is directionless on a generation by generation scale. Sometimes it will increase the frequency of P, sometimes it will decrease it. This means that on average, especially over short time intervals, the net change is zero. So what did these researchers find? In a paper published in 2024 in the Proceedings of the National Academy of Sciences, they report that the average change of allele frequencies over their 10-year study was 0. There didn't appear to be any directional shifts in allele frequencies. This is exactly what you would expect if the only evolutionary mechanism at play was genetic drift. So, is the population just evolving randomly? Not exactly. While the average change was 0, the researchers found that the variance each generation was very large, much larger than what you would expect by genetic drift alone. It was simply that in one generation, the allele frequency would shoot up, and in the next, it would shoot down in these really big swings. Averaged over time, the change is still 0, even if over one time interval, the shift is quite large. This implies that natural selection must be at work, as the allele frequency change is greater than what would be expected by pure chance. So which is the correct interpretation? Is it genetic drift or natural selection? Instinctively, most of us might be tempted to say it's still natural selection. All that is happening is there's variation over time in the direction of selection. For example, maybe for individuals born in the spring, selection is favoring alleles that boost immune response to certain parasitic fungi that bloom in the spring. But in the winter, those same alleles maybe reduce longevity, and so are disfavored since the fungi are absent in the winter months. This would lead to seasonal shifts in allele frequencies. This has sometimes been called fluctuating selection, and the earliest cases of this go back to the 1940s with the work of Theodosius Dobzhansky, who observed these seasonal shifts in natural populations of fruit flies. In reality, the answer is a bit more subtle than this. In early population genetic models that assumed infinite populations, fluctuating selection actually maintains both alleles. They just fluctuate around some average frequency. The magnitude of their fluctuations is determined by how large the strength of selection is over time. Very small shifts lead to minor deviations from the average, whereas strong shifts cause really big swings. But ultimately, so long as the population is infinite, both alleles stick around. This is obviously different from a random process like drift that will eventually cause one of the alleles to be lost and the other to be fixed, irrespective of the effects of those alleles. But what if the population is finite instead of infinite, as all real populations are? To answer this question, let's take a quick journey back to the 1950s. Enter stage right. The British geneticists R.A. Fisher and E.B. Ford are studying a population of the scarlet tiger moth. They track how different genotypes shift in the population over time, much like our Daphnia researchers, and they find that generation by generation shifts are very large, much larger than can be explained by chance. Fisher believes these findings are a vindication of a theory that he proposed over 20 years earlier, that natural selection can itself maintain genetic diversity in a population, either by favoring heterozygotes or by fluctuating back and forth through time. Now enter stage left, the American geneticist Sewell Wright. Wright rebutted Fisher's claim by pointing out that Fisher hadn't actually ruled out the possibility that genetic drift was responsible for these large shifts. With some fancy math, he showed that Fisher's conclusions were premature, that there were many ways to get the exact same shifts that Fisher had observed. Now, in our audience was a young, up-and-coming Japanese population geneticist named Motoo Kimura. Kimura asked himself, if Fisher is correct that the allele frequency in the moths is fluctuating due to selection around an average effect of 0, would both alleles still be maintained in a finite population? After all, genetic drift also produces an average frequency change of 0, but ultimately one of the alleles will eventually be lost. While on board the passenger vessel Hikawa Maru on his way to America to start his graduate degree, he worked out the mathematical details to this fascinating problem. What he discovered was quite remarkable. He found that whenever natural selection has a net effect of 0, that is, that it fluctuates relatively equally back and forth, favoring and disfavoring the same allele, it behaves as what he called quasi-neutral. Like a neutral allele, it had not only a predictable fluctuation each generation, but even a predictable rate of fixation. While the rate of fixation of a neutral allele is 1/2n, the rate for a quasi-neutral allele is v sub s over 8, where v sub s is the variance in the direction of selection each generation. For example, if in one generation selection favors an allele by 5%, then the rate of fixation is 0.05 over 8, which is equal to 0.00625. For comparison, for genetic drift to fix an allele at this rate, it would require that the population size, n, is only 80 individuals. This tells us something fascinating, that when natural selection acts like a random variable, fluctuating around a mean effect of 0, if it has a large fluctuation, it will lead to a rather rapid fixation of one allele or the other, irrespective of the fact that it is favoring neither of them consistently through time, and that which one fixes is totally random. Fast forward back to our Daphnia study from 2024. The researchers knew about Kimura's mathematical model and applied it to their water fleas to ask the question, how might fluctuating natural selection impact how much genetic diversity our population should have? To answer this, they modeled a range of population sizes, from 10,000 all the way up to 10 million, and a range of selection strengths from very weak fluctuating selection at 10e to the -6 to very strong fluctuating selection at 0.01. They compared the results to how much neutral genetic diversity a population is expected to harbor under mutation drift equilibrium. That is, where all the diversity that exists is solely the result of a balance between mutation adding new alleles and drift causing them to be lost. What they found was startling. When fluctuating selection is very weak, irrespective of the population size, their model showed that populations should have as much genetic diversity as expected under mutation drift equilibrium. So far, so good. But when the selective variance was large, there developed a negative relationship between population size and genetic diversity. This is quite counterintuitive. It's textbook knowledge that larger populations have more diversity, right? It turns out that this is only true if those genetic variants are truly neutral, as opposed to quasi-neutral. Quasi-neutral alleles, as Kimura showed in 1953, are lost much faster than truly neutral ones, so genetic diversity is rapidly depleted in large populations. In fact, our Daphnia researchers found that it's not only quasi-neutral alleles that are lost at a more rapid rate when the population gets large in size. It's also the truly neutral alleles that happen to be linked to them due to physical proximity on the chromosome. Thus, for very large population sizes with strong fluctuating selection, the amount of genetic diversity is much less than expected at mutation drift equilibrium. These findings have major implications for our expectations of how much genetic diversity a population is expected to have. In 1974, Richard Lewontin pointed out that genetic diversity doesn't appear to scale in a one-to-one fashion with population size, as would be expected if population size alone is the sole determinant of diversity. This is the prediction under Motoo Kimura's neutral theory, which states that genetic diversity should be equal to the product of 4n mu, where mu is the mutation rate and n is the population size. Instead, at larger population sizes, Lewontin noticed that diversity appeared to flatline, what became known as Lewontin's paradox. But if fluctuating selection is common in nature, and there are many of these quasi-neutral alleles, then it would beautifully explain why large populations have so much less diversity than you would expect under strict neutrality. And yet it's not because of consistent directional selection, as many adaptationists have claimed, but instead the result of selection behaving like a random factor. This example of how selection can behave randomly is from the bottom up. At the level of the generation, selection is deterministic, favoring one allele over another. But at the top level, averaged over time, it generates a pattern of randomness, only different from genetic drift, and that the random fixation of one allele or the other is faster than under drift alone. This bottom-up randomness is like our chess example. At the level of the chess game, the bottom, the outcome is deterministic. And yet at the level of all 100 games, at the top, the pattern is one of randomness. Hopefully I've convinced you that this is not merely a semantic quibble, but results in fundamentally different outcomes, sometimes with counterintuitive results. Now, let's reverse the direction and talk about how deterministic selection at higher levels can result in randomness at lower ones. As you emerge from the expanse of marshlands and bayous of the Mississippi Delta in southern Louisiana, following I-90, you'll cross this massive bridge over Bay St. Louis and find yourself in a sleepy little coastal town called Pass Christian. The locals sometimes call it the Pass. When I was in graduate school, I went to the local beach there to see if I could dig up shovel bugs, small amphipod crustaceans that live on sandy beaches. My graduate thesis was on the population genetics of several species of shovel bugs, and I was hoping to find a healthy population on the eastern side of the Mississippi River for DNA sequencing. As luck would have it, there was a vibrant population there. I scooped up a dozen or so, preserved them in ethanol, and a few months later, sat down at the lab computer to analyze the DNA sequences that we'd just gotten back from the sequencing corps. What I found was puzzling. You see, All the samples that I'd collected from the past were the same species, Lepidactylus triarticulatus. They have several morphological characteristics that make them just unmistakable, so I was sure that I hadn't misidentified them. And yet what the DNA sequences seemed to suggest was that on this beach, there was not one, but two species. That is, the sequences were so diverged from each other, they couldn't possibly belong to the same interbreeding population. I knew there wasn't a problem with the DNA sequencing quality. I was doing Sanger sequencing, a bit outdated, but the benefit is that you can check the readout of each and every nucleotide and confirm that the chromatogram gives a nice clean peak. So I went back and I checked the specimens. Maybe I'd missed something. Nope. They were definitely Lepidactylus triarticulatus. But I still wasn't convinced. If these sequences were correct, it would imply that there were two morphologically indistinguishable species living on the same beach. Just ignoring the ecological principle of competitive exclusion, what would be preventing them from interbreeding? It just didn't add up. Another student in the lab was driving to Florida in the next month, so I asked them to stop by Past Christian and grab me some more specimens. Surely what I'd gotten was a fluke. But when I got the sequences back from the second collection, it was clear. There were two different species on this beach, and they both looked just like Lepidactylus triarticulatus. When you have two species that are morphologically identical, we designate them as cryptic species. In amphipods, this phenomenon is actually quite common, but it's rare that you find two cryptic species living in the same area. As I did more DNA sequencing, I found that Lepidactylus triarticulatus was an entire cryptic species complex, and that the second species on Past Christian was a more recent arrival from west of the Mississippi. The existence of cryptic species implies something curious. Populations can diverge from one another genetically, but not morphologically. An obvious question that arises from this is, why not? Especially if populations are geographically isolated, wouldn't they adapt to each environment differently and thus diverge phenotypically? Not necessarily. You see, all shovel bugs live in virtually the same uniform habitat, highly oxygenated, wave-exposed sandy beaches. No beach is identical, obviously, but the trials you face on one are very similar to what you'd face on another. And shovel bugs are extremely well adapted to this lifestyle. They have highly modified each of their legs compared to other amphipods, broadly expanding them and elaborating them with spines and fluffy quills. Their front legs, as their common name implies, have been shaped by natural selection to act like little shovels, which enable them to rapidly dig into the sand. If anything, we might expect natural selection is actually preventing these populations from changing. When natural selection acts in this way, we call it stabilizing selection. In effect, stabilizing selection acts to purge phenotypes that deviate too far from the optimum. But if stabilizing selection is preventing morphological change, how can there be genetic change? A simple way is that the genes that are changing are simply not contributing to the trait that stabilizing selection is acting upon. So genetic divergence in these populations is just due to drift. But we want to know specifically about those genes that are changing that do underlie the trait. The first thing we should bear in mind is that traits are rarely the result of a single gene like in simple presentations of Mendel's laws. They are normally determined by dozens or even thousands of individual loci, each of which may have multiple alleles in the population. For example, we can imagine a trait like the number of spines on the leg of a shovel bug as being controlled by 10 genes. At each of these genes, there are two alleles. We'll call them 0 and 1/2. If you have the 1/2 allele, you get half a spine. The 0 allele gets you no spine. So you need to have at least two copies of a 1/2 allele at any locus to get a full spine. Let's say the optimum number of spines in the environment is 5. Too few and you don't have the necessary defenses. Too many and you don't move efficiently through the sand. An example genotype might look like this. Each of the different parentheses are the loci. As you see, there are 10 of them, and the two numbers within that loci represent the alleles that you have. So this genotype has 3 loci that are heterozygous, which means they have 1 full spine and 1 half spine from those heterozygous loci. Then they have 4 loci that are homozygous for the large spine, and 3 that are homozygous for no spine. Thus, this shovel bug would have 5 full spines and 1 half spine on their leg. They would thus be slightly less fit from the optimum of 5, but they're not far off. Notice how this scheme lends itself to getting 5 spines in a multitude of ways. But so long as there remains genetic variation in the population, individuals can be born with suboptimal combinations. To see this, imagine a shovel bug that is homozygous for every locus, and their genotype is this. Notice how this would guarantee that they have exactly 5 spines, and if they mated with someone that had their exact same genotype, all of their offspring would also be guaranteed to have 5 spines. Of course, this only works if their mate has exactly their genotype. If they are homozygous for a different combination, say something like this one, then their offspring would end up being heterozygous for every single locus. Summing up their allelic contribution, we see that their offspring indeed have 5 spines and so have the optimum number, but the issue arises as soon as they try to find a mate. If they mate with another fully heterozygous individual, for example, then many of their offspring would have combinations that would deviate from the optimum. Thus, stabilizing selection is acting to remove genetic variation, but it's doing so in a particular way. It needs to lose and fix the same alleles. But again, it doesn't matter which ones, so long as they are the same. And yet, despite this, stabilizing selection does appear to have an odd preference for which one it removes. To understand this, let's rewind the clock back to the 1950s one more time. Now we're in the Institute of Animal Genetics in Edinburgh, and a pair of animal geneticists named Douglas Falconer and Alan Robertson have just done a series of breeding experiments on mice. They're interested in how selection for body size in mice might differ between mice at the mean of the distribution versus those that have extreme phenotypes. At that time, there was this idea that stabilizing selection might maintain genetic diversity in the population. The thinking was that individuals near the center of the distribution might be highly heterozygous, whereas those at the ends might be homozygous but for the wrong alleles, soon to be purged. Thus, becoming more homozygous would push you towards the extremes, and stabilizing selection might be acting to preserve the diversity at the center of the distribution. But what they found was the opposite. When they selected mice from the center of the distribution, it led to the rapid fixation of alleles and reduction of the variation around the mean. This meant that by artificially narrowing the distribution, they actually strengthened stabilizing selection. This revealed that the direction of stabilizing selection was not toward preserving genetic diversity, but towards purging it. Alternatively, when they bred mice at the ends of the distribution, genetic diversity was actually maintained in the way you'd expect under a model of negative assortative mating. All the alleles eventually reached a stable equilibrium frequency. Robertson realized that there was no theoretical explanation for this, and so he sat down to see if he could work out the algebra to explain it. In 1956, he published an extremely important paper that revealed mathematically that the action of stabilizing selection is to purge what is called the minor allele. That is, whatever allele at that locus is at the lower frequency. How fast is the minor allele lost? It turns out the rate of loss is proportional to the square of its effect size. So for alleles with very tiny effects, the rate of loss is extremely slow. If the effect of the allele is quite large, then stabilizing selection purges it rapidly, much faster than neutral drift. But again, selection doesn't see which allele it's purging. All it sees is the phenotype. It's not that the major allele is better than the minor one. Let's return to our populations of cryptic shovel bugs to understand the implications of this. In effect, we can imagine each population as a kind of replicate. They all have the same alleles, but starting at different frequencies based on when that population was founded and what the source population's allele frequencies were. Stabilizing selection is acting on exactly the same traits in exactly the same way, but since their frequencies aren't the same, which alleles end up getting fixed or lost is random across populations. Thus, all populations look the same morphologically, but they have fixed and lost different alleles that contribute to the phenotype because there are just many ways to get that exact same phenotype. Another way to think about it is that selection is acting at the higher level on the phenotype, while at the lower level, the genotypic level, alleles are being randomly and differentially fixed across the separated populations. Hopefully the pieces are now in place for you to see how this process of deterministic selection at the level of the phenotype and more or less random selection at the level of the genotype can actually drive speciation while keeping each species morphologically identical. To make it clear, imagine we have two populations of shovelbug, one that has been on the past Christian beach for a long time and one on the other side of the Mississippi River. Stabilizing selection keeps the populations the same, but they randomly fix different alleles. Here's the different genotypes that they have been fixed for. All good. Both have an optimum of 5 spines. Now we have a chance migration event. A few of the shovel bugs happen to get swept up into the current and they make their way across the river to the past Christian beach. They can either mate with their fellow migrants or with the resident population. Obviously, if they mate with the migrants, all their offspring have the optimum number. But what about mating with the residents? Just as before, the first generation would be fully heterozygous and thus be safe from stabilizing selection. But in the F2 generation, when those hybrids start to mate with either parental population, mismatches begin to occur. Their offspring mostly deviate from the optimum and thus are selected against. Hence, hybrids end up having lower fitness than either pure mating populations do. Over time, stabilizing selection acts to purge the minor parental ancestry, keeping these two cryptic species genetically distinct from one another. If shovelbugs feel a little removed from your daily life, you might be interested to know that if you are of European or Asian descent, this exact same process is happening in your genome. In a paper published in March of this year, in 2025 in PLOS Genetics, evidence was presented that stabilizing selection at functional genomic positions was purging Neanderthal alleles in the human genome, but also that human alleles were apparently being purged from the Neanderthal genome as well. Instead of arguing that humans and Neanderthals have some functional divergence at these loci, the author makes the argument that humans and Neanderthals have merely fixed different alleles that ultimately contribute to the exact same phenotype. We have now seen two ways in which natural selection, a deterministic process, can induce randomness at different biological levels. It can drive allele frequencies each generation, and yet if it's tracking random changes in the environment, it can generate an overall pattern that resembles just a sped up version of genetic drift. It can also act at a higher level on individuals themselves and even across multiple populations, and yet permit stochastic loss and fixation at the allelic level that can ultimately cause populations to be incompatible despite having the exact same phenotypes. Thanks so much for being here. I hope you found this interesting and illuminating, and the next time you hear someone say that selection is deterministic, you can be the, well, actually, person in the room. If you like this video, hit like and subscribe, leave me a comment, and share it with your friends. Until next time, take care.